this morning is just an opportunity to continue what I see as like building relationships between CHOP, Penn, and UBC. And I'm really delighted to just share some of the work that we've been doing over really the last decade and see how it aligns with a lot of the work in the investigation in adult kidney stone disease. So as a way of disclosure, the research that is going to be presented here was funded by NIH and the Frontier program, which is an internal mechanism at CHOP. I am on the scientific advisory boards for a couple of pharmaceutical companies. None of those relationships have anything to do with the work presented here. So the objectives are threefold. One is to understand the association between antibiotic exposure and kidney stones, and then to move into the underlying likely causal pathways between those associations in early onset stone disease, focusing on the microbiome and metabolome, and then its similarities with the work that Dr. Chu and Dr. Lang have done with stone disease among adults. And then finally, to move into the space where we're moving, which is the proteome and metabolome, to identify and discover new diagnostic tests for stone disease across the lifespan. So this is our really conceptual model of stone disease, which I think even in medicine and the lay press, stones are understood as a condition that's extremely painful, but episodic. While that's true, it doesn't really describe the severity of the condition. It's really a disorder of mineral metabolism. And that disorder is manifested by these acute episodic events, but it also leads to conditions such as chronic kidney disease, loss of bone mineral density, leading to increased risk of fracture, hypertension, coronary disease, et cetera. And when we're talking about individuals who form stones early in life, which we define as under the age of 26 years, all of those conditions may be a greater risk because there's a longer lifespan over which they may occur. And because when stone disease, as with many other disease, starts early in life, it may represent a more severe condition. So that's our driving force behind this research, is to not just understand those associations, but to understand the causal mechanisms so we can intervene early and prevent these painful events, but also the end organ effects of stone disease. And we focus on the exposome and its interaction with the gut-kidney axis, which we'll get to in about 5 to 10 minutes. So why do we do this work? Why do we focus on early onset stone disease? Historically, stones was a disease of white middle-aged men. There used to be a 3-to-1 male to female predominance. And over the last 20 to 30 years, we've seen an increase in the prevalence, the number of individuals living with stone disease, that's doubled. And while that has happened in all age groups, what has really driven that increased prevalence is the rising incidence in females, both young females and particularly adolescent girls, where the greatest rise has been observed early in life. So we're seeing a rapid rise of stone disease under the age of 26. And in those individuals, females are much higher than males with stones. So that has caused a shrinking and really elimination of that historic gender gap and a new population of patients for whom we have very little evidence to improve their care and to improve outcomes for individuals living with the disease. So referencing that possibility that stones, when they occur earlier in life, may represent a more severe condition, we know from some historic work that once a symptomatic event occurs, the probability of having another symptomatic event is about 50% within that first 3 to 5 years of the index stone. This is at least as high as individuals with stone disease later in life, if not higher. So as we've seen this rising incidence of stones in youth, it has created really two problems. One, that the evidence that we have supporting management of children and adolescents and young adults with stones is incredibly weak, because we've never had any need to have large-scale clinical trials and to have dedicated efforts to develop new diagnostics and therapeutics for these children, because they simply haven't been affected by the condition. Now they are. And then broadly across stone disease, advances in diagnostics and therapeutics have stalled. We're still using thiazide, we're still using citrate for a condition that affects about 11% of Americans and many more globally. It's hard for me to think of any other condition that affects so many and impacts their lives in so many ways, for which we have very few diagnostics and therapeutics. So the key questions that drive our research is, number one, why is stone disease beginning earlier in life? And then number two, how can we translate this knowledge into new therapeutic and diagnostic discoveries that improve outcomes, not only for children, but really for individuals across the lifespan? Stone disease is the same in children as it is adults. It's just a different manifestation at a different point in time. But the causal pathways are likely very, very similar or the same. So we talked yesterday about intention and opportunities in research, and this is really the beginning of the journey for our road to understand the gut-kidney axis. And this, the sign on the left was something that I took back in 2016 as I left the train station in London and walked under St. Mary's Hospital. And this is the laboratory where Sir Alexander Fleming discovered penicillin. And that marker just happened to coincide with the same time that I was reading this book by Martin Blaser, who's at NYU, about missing microbes. And he focused on H. pylori. But it was really the beginning of this broader conversation of how the gut microbiome could impact health. And if you are missing certain organisms, how those could be keystone species that could lead to diseases that he called modern epidemics. And he focused on ulcer disease, gastric ulcer disease, but we focus on stones, which at that time we thought had a similar sort of framework for understanding the disease. So this is our conceptual model that we've developed over the last 10 years for the gut-kidney axis, where on the left side you have the exposome, which is like the total amount of exposures that we have in our lives, whether it's diet, antibiotics, environmental toxins, of which we may or may not be aware, and how those exposures impact the composition of the gut microbiome, and in turn influence the composition of the intestinal metabolome, and how those may manifest downstream in the end organ in the kidney with changes in urine metabolites, as well as urine chemistries. But if you look at this conceptual model, the only thing that we really focus on currently is urine chemistries, which we suspect is a small part of this overall pathway that ultimately leads to kidney stone disease. So we wanted to start much more proximal in this pathway to understand how perturbations of the gut microbiome may ultimately manifest in stone disease, presenting new opportunities to work upstream of the kidney. So our initial venture along this axis really came out of that experience in London when we hypothesized that exposure to antibiotics may be associated with kidney stones. And to investigate and answer this question, we used a large database, which at that time was called THIN. It's a UK-based database in which they capture all care delivered in the UK. And because it's all delivered through the GP, you have really complete capture of exposures, pharmaceutical exposures, as well as disease for the entire population. So this was a case control study in which we had about 26,000 individuals across the lifespan who had kidney stone disease that were then matched with a 10-to-1 ratio. So it was like 260,000 individuals without stone disease. And we matched on age, sex, and GP practice. to look for differences in prior exposure to antibiotics. And what we discovered, no matter how we modeled this association, was that exposure to 5 different classes of antibiotics was associated with an increased odds of kidney stone presentations and kidney stone disease. And those classes of antibiotics were cephalosporins, fluoroquinolones, nitrofurantoin, broad-spectrum penicillins, and metronidazole. Or I'm sorry, it's actually shifted up. It should be sulfa rather than metronidazole. So in this, there were some individuals that had UTI, but the most common indications were for like upper respiratory infections, pneumonia, and some skin disorders. So this led us to begin to ask the question of, if there is an association between antibiotics and stones, does it matter when this exposure occurred? So when these antibiotics were given earlier in life, the odds of stones were greater in all the different classes of antibiotics. Now, the magnitude of that association differed. It may have been greater for fluoroquinolones than it was for nitrofurantoin, but that trend and the shape was the same. So when the exposure occurred earlier, the odds of stones were greater. And we don't have we don't know the mechanism here, but we can postulate. So if you are exposed to antibiotics earlier in life, when the gut microbiome is developing and may be perhaps more vulnerable to insults or exposure, you may cause a shift that depletes your bank of all of the factors that protect against stones. So this consistency was consistent with our guiding hypothesis. And what we also understood from this study was that once an antibiotic exposure occurred, the risk for those 5 classes of antibiotics was elevated. It was greatest within the year following the exposure, but it never went to 0. So even years down the road, we started to decrease.
Can I ask a question about that related to your earlier slide too? Is the, cephalosporins, penicillins have similar sort of spectra of activity, and yet, different risk factors. How do you explain this correlation based upon target sort of bacteria?
Yeah, that's really where Dirk and I want to go next, is to understand how these individual exposures change the composition of the microbiome as it relates to stone disease. Right now, we don't have like that causal pathway identified. We don't know the impact on what I would say communities of organisms as they relate to stones. We have these like broad associations, again, through very different mechanisms, but it's an unanswered question to this point. We have the opportunity to, I think, ask and potentially answer that question, which I'll get to at the end of the talk, but unfortunately can't provide an answer now.
And is this like any exposure to antibiotics, or is there a difference between treatment versus?
Great question. So this was a massive database, but it still wasn't large enough to answer some of those important questions, such as the duration of exposure, the strength of the exposure, prophylaxis or treatment dose, and the interaction between like multiple exposures of the same antibiotic or exposures of different classes of antibiotics. We've just completed analysis using a large claims database called HIRD, which is Anthem Insurance in the US, that has answered those questions. It does look like there is some interaction between antibiotics, and we're still reviewing the data for prophylaxis, but we hope to be able to provide that with that. So as we moved on from that study in 2018, colleagues at Harvard, and this is Gary Curhan's group, published a study in AJKD using the Nurses' Health Studies 1 and 2. And they found very similar associations in women who were exposed to antibiotics with incident stone disease. So the strength of Nurses' Health is that they have it's a prospective cohort study, and with new onset disease, they're able to identify that And through some of the questionnaires they administer, they're able to understand these prior exposures. So this strengthens the study design then from what we did, because a prospective cohort study compared to a case control, you're able to have more causal inference. But some of the limitations of it was that they didn't have the classes of antibiotics. It was just simply antibiotics, yes or no, over a certain time period. But the consistency of these point estimates, 1.5, 1.4, are very similar to what we identified in a different population. And so why is this a potential concern? So the populations who are exposed to antibiotics at the highest rate are women and children. And these are the groups who we know that stones are increasing at the fastest rate. This is all ecological, and it certainly could be ecological fallacy. We're seeing like trends in the same direction. Are they related? We don't know. But it certainly, I think, is a question worth asking. And that moved us into really the investigation of the causal pathway. So we have these broad associations. We don't have an explanation for, what impact these antibiotics are having on the gut-kidney axis. We don't really know those differences at this point between individuals who have stones and those that don't, which is what prompted this next sort of phase of research. And this was, I think, going off of some of the questions yesterday of like, how does an English major who's a urologist and a clinical trialist know anything about the microbiome? You're not a microbiologist, and all of that is true. It was true then and it's true now. It's really through partnerships and developing collaborations. And having experts come together that each inform a question. And I think I also referenced yesterday that so much of this work has been accelerated from a conversation that Dr. Lang and I had in a Paris airport so many years ago from World Congress. And it's those, I think, relationships where you can have experts inform an idea and then identify ways to answer that question through unique contributions that I think gives us the opportunity as urologists to really do meaningful work. And at this time, now about 7 years ago, a lot of the focus globally on the microbiome was on the diversity of the microbiome, and then in stone disease on this particular organism called Oxalobacter formigenes, which has a lot there's a lot of rationale for focusing on Oxalobacter because it degrades oxalate. And it uses oxalate as its carbon and energy source. And there have been studies that have shown that if for individuals with stones, they have a lower prevalence of Oxalobacter in the gut. And there's a lot of biologic plausibility. But we were thinking that when you look at one organism, you're really not thinking about these communities that exist in the gut and elsewhere, just like humans. They're not single entities, but they exist in relationship to others. So we wanted to take a step back from the focus on Oxalobacter to go a little bit broader. And some of the work that inspired the research was done by a group in Italy, Tasian, who published a study of adult stone formers with unknown composition. We know these were radiopaque stones, but nothing more about them. That showed that the diversity of the gut microbiome of these individuals with stones was lower than controls, those who don't have stones. They found no differences between Oxalobacter between these two groups, but they did find some differences in oxalate degrading genes between these two groups, and that there was an association with lower urinary oxalate in the controls than the individuals with stones. So this provided some background for us to ask some different questions. So all the prior studies had been done in adults with kidney stone disease. There was no information about those with early onset disease, which is what prompted this study. And again, this was a case control study of what were 88 individuals, 44 of whom had calcium oxalate stones with at least 80% of the stone made of calcium oxalate. These were all individuals under the age of 18 at time of stone diagnosis who had idiopathic stones. They were otherwise healthy, which is really the patients we see. We see, healthy 14-year-old girls who don't have IBD. They don't have any other serious health conditions that may be more common in adults. And for this study, we matched them with 44 controls who are alike in every way: age, sex, race, ethnicity, time of the year, except that they didn't have stones. So we want to make these groups as alike as possible. So each of these participants underwent extensive phenotyping about their past history. We also administered dietary recalls to each of these participants 3 times. Which has some advantage over food frequency questionnaires and allows you to understand the typical diet of these participants. They also provided a single stool sample that was analyzed using shotgun metagenomics, as well as untargeted and then later targeted metabolomics. We did not obtain urine samples for these participants, which certainly was a missed opportunity. So this is the heat map of all of those 88 participants. You see on the left in blue the control participants, on the right in the salmon color the cases. And when you look at this high 30,000 foot view level, there's much more similarity than there are differences. So broadly, it looks like the composition of the microbiome with each row representing a taxa is more similar than it is different. But when you start looking at the differential abundance of bacteria between these groups, there were 31 bacterial taxa that were different between those with early onset calcium oxalate stone disease and their matched counterparts. And each of those 31 bacteria are shown on the left-hand figure, and each of those bacteria was lower among controls than they were cases. And at this time, our hypothesis was that we would see a lower prevalence of oxalate degrading bacteria in those individuals with early onset stone disease, which we did. We saw no differences in Oxalobacter formigenes. It was an incredibly rare bacteria between these groups. But what was surprising, and we did not anticipate going in, was that the stronger signal was in butyrate producers. So we identified these 7 different taxa, and now there's more compared to what we knew about these bacteria back then. That produce butyrate, which is a short-chain fatty acid that helps maintain the gut mucosal barrier. It directly regulates some of the oxalate transporters in the gut and the kidney, and has been involved in inflammation in the gut and the kidney. So this was a really, I think, intriguing finding that opened up new avenues of investigation and our understanding of the potential causal pathways in the gut-kidney axis of stone disease. With that shotgun metagenomics, we had a way of estimating the expression, not the expression, but the presence of genes that are involved in both butyrate production and oxalate degradation. There were no difference in the genes in oxalate degradation, and there were some differences in the genes involved in butyrate production. The one that was statistically significant after correcting for all of the false discovery was butyryl-CoA dehydrogenase, which you see in the top figure on the right. And I think these findings gave strength to our growing belief that if you focus on one bacteria, not just one type of bacteria that does certain functions, but one bacteria, namely Oxalobacter, you're simply missing the forest for the trees. And I think it's necessary to step back and be open to intriguing but as yet unexplained results that may move your research in new directions. So when we recognized that Oxalobacter formigenes was not a part of the story in at least these 88 individuals that we think are representative of early onset stone disease, we found that oxalate degradation is an important part of this pathway, but it's really a network of bacteria, of which Oxalobacter formigenes may be a part, but a minor component. It's really many other bacteria that have relationships with each other that perform this common function. And this is just the map, the network of those bacteria that we identified in these groups. In these 88 participants, we also performed untargeted metabolomics in the stool sample. And that gave us the ability to identify the metabolites that were different between the groups. And unlike the composition of the microbiome, where all of the bacteria were lower in controls, here we found that there were some metabolites that were higher in the healthy individuals and some that were higher in the cases. So there were 18 metabolites that were different between the groups, 10 of which were higher among the healthy individuals, and those were mainly around amino acids. The 8 metabolites that were lower among cases were in the lipid family. We don't have more information about the specific types of lipids because we did not anticipate at the beginning of this study that short-chain fatty acids would be a part of this story. We didn't store the stool specimens in a way that would allow those analyses. These short-chain fatty acids are volatile, and unless they're stored properly, you can't analyze the presence or the abundance of the short-chain fatty acids, of which butyrate is one. So again, that was, I think, perhaps a missed opportunity if we had looked beyond stone disease to understand how short-chain fatty acids are so important in other diseases, including IBD. So reading outside the urologic literature was a lesson learned that for me at that time.
Yeah, I guess this is my question relates to that, and that's stool prep storage, even for your essentially their transcriptome analyses. Is that right? So for your transcriptome analyses, one would imagine that relatively small differences in storage will lead to very differences in composition. So how closely do you standardize stool collection?
It's extremely standardized. It is done. We got our protocol from the Penn Chop Microbiome Center, which started their work in early onset IBD with a lot of home collections of both cases and controls. So all the participants, save one, collected the stool specimen at home. It was put in a shipping container with cooling packs, and then with same-day delivery through FedEx, received by our center. It was immediately aliquoted and then stored at -80. So there certainly are batch effects, which we want to limit in the analyses. So rather than analyze these samples as they came in, they were all analyzed together at both the sequencing level and with the untargeted metabolomics to minimize those batch effects, which we know occur. So they were all standardized. They were collected in the same way. But you ask a really important question of the methods of collection, storage, and analysis. And one of our sort of guiding principles is no freeze-thaw cycles. And if they are done, then they're done on all samples in the same way. And one point to clarify, we did not perform transcriptomics, which I think is very important. It's also very expensive. Dr. Nizal, who's a nephrologist at NYU, has done some really intriguing work with transcriptomics in hyperoxaluria and have found really profound differences in the expression of genes compared to the presence of genes. So the active transcription of these genes may be a really important part of the story of which we're just beginning to understand. And one thing that we had the opportunity to do in this sample, which ranged from age 4 to 18, was to look for differences in the diversity of the microbiome by age. we knew, or we suspected and confirmed, that there would be a lower diversity of the microbiome, particularly alpha diversity, in those with stone disease compared to controls. That wasn't an essential part of the story that we wanted to tell. What we asked was, are there differences in the diversity of the microbiome based on when stones occurred? And by comparing the diversity of the microbiome by age at study participation, which reflected age of disease diagnosis for control or for cases, we found that there was an age dependence of the diversity of the microbiome, where it hit a nadir about 10 to 12 years. And for cases, there were no differences. So no matter if you were a 4-year-old, a 12-year-old, or an 18-year-old, and you were healthy, the diversity of the microbiome was the same. But there was a lot of difference among those with stones. And hitting that peak of low diversity at 10 to 12 years is an interesting finding for us, because that's the time at which the incidence of stones really starts rising among children and adolescents. So we see this emergence in early adolescence. So if you're seeing this low diversity immediately preceding that, it's possible, and again, unproven, that this could be a part of the causal pathway of early onset stone disease. So the correlates in adult disease. And this goes back to the conversations with Dr. Lang and his group, and I will in no way try to summarize it in the detail that it deserves, but I wanted to point out some of the really, I think, striking correlates between early onset disease and adult disease. And this was from a study, Dirk, this was back in 2020, was it? So this was in Kidney International, and looking at the bacterial networks between adults with stone disease and those without. And I'm going to read this quote. Interestingly, Oxalobacter was conspicuously absent from the list of OTUs enriched in controls, suggesting that other bacterial species are more important to prevent urinary stone disease and or oxalate homeostasis. In addition, we found that the number of co-occurrence interactions with bacteria associated with Oxalobacter discriminated patients from healthy controls more effectively than looking at the presence or absence of Oxalobacter formigenes alone. That sentence could have been written not as eloquently as Dr. Lang and colleagues did in our paper. It is not just about Oxalobacter formigenes. It's about these network of bacteria that may work synergistically that can differentiate those with stones at any age of life. And this is one of my favorite papers that I've read this year, that was published in 2024 in Urolithiasis, again with Dr. Chu and Dr. Lang, looking at butyrate metabolism in the gut of individuals with stones of different compositions compared to controls. And what was remarkable to me was that in this very diverse sample, of which there were not many, and I view that as a strength. So when you look at that you're seeing such profound differences with such a diverse sample in few participants, the magnitude of those differences has to be really high to detect. So I view that you have such profound differences in a relatively small sample as a strength, given its consistency with what we know. And these differences in the specific bacterial taxa and their functions are completely aligned with what we're seeing in early onset stone disease, in that butyrate production is a key, if not the key, part of this causal pathway. And the more that we can align efforts to really consider stone disease a disease across the lifespan, and not one that is unique to children or unique to adults, but has common features in which it's important to identify differences, I think the more impact this type of research will ultimately have. So I've learned a tremendous amount from reading these papers. So where are we going? So where we are going is the final phase of the Pursuing Optimal Organisms in People with Stones, or the POOP study. I cannot take credit for this name. This is all Justin Ziemba, who's a very close colleague and endourologist at Penn, who has a unique skill for developing acronyms. So what the POOP study does, and UBC was just a key partner in this study, is expand our sample size from 88 to 300 and really go for disease across the lifespan. So with a minimum age of 4 and no maximum age limit, we have collected nearly, at this point, 220 samples from individuals, cases, and controls, and are in that final phase of getting to 300 now. So what POOPs will do is collect the stool samples, which we have stored in a way that we can do targeted metabolomics, and this will be short-chain fatty acids as well as potentially bile acids, depending on the amount of funding, but also urine samples. So those urine samples, we have spot urine samples from each of these participants, as well as 24-hour collections, which Dr. Asplin was kind enough to save a small sample of that 24-hour collection so we can have a referent for the spot urine sample. The goal of spot urine samples, which we'll get to, is to move from a test that is laborious and very painful for patients to collect into one that can be done at the point of care, either at home or in the clinic. So we're still focused on this causal pathway and are building the infrastructure to be able to, I think, make some advancements here. By collecting urine, we are focusing on urine metabolites, which we hope and expect will expand the spectrum of certain aspects of the urine that can be leveraged into developing new diagnostic tests. My personal belief, and I have the greatest respect and admiration for Dr. Asplin and what he's accomplished, is that urine chemistries are just such a small part of this pie. And if we expand it from here to even here, we're going to have a much greater ability to identify some potential therapeutic targets, but also be able to identify exacerbations of disease before they occur. So by going from chemistries, calcium, oxalate, pH, sodium, into these urinary metabolites, which we're beginning to scratch the surface, I think we'll have the ability to deliver care in a much faster, much more precise way. And here are some preliminary data. I have to show it in aggregate just for IP reasons, but these next slides demonstrate some preliminary data of the work in proteomics and metabolomics using these urine samples from a subset of our patient and controls. So a panel of these 10 urinary proteins, and these are all done on spot urine samples, has a high ability to discriminate between individuals who form stones and those who are healthy counterparts. When we move into metabolites, which we view as complementary but different than proteomics, we see in this volcano plot, again, a strong ability to discriminate controls from individuals with early onset stone disease. But that, to us, is not the key question. We know who has stones. We know who don't have stones. That is an initial start, but what we really need to do is, among stone formers, who has severe disease that's most likely to progress, most likely to recur, most likely to have a manifestation before it appears on imaging, or especially before it appears as a recurrent stone event or in the metabolic manifestations of the disease. So what you see here are plots of the, So you see plots of the urinary metabolites where it differentiates based on the abundance of these metabolites, the presence or absence of severe disease. And because we collected these samples across a lifespan, the way that we classified on the left is that if you have early onset disease before the age of 26, compared to later onset disease, those with early onset disease were classified as severe stone formers. In that we identified these metabolites that were both overly expressed on the right and underexpressed on the left side of the plot, that discriminated those with severe disease from those with what we consider routine or routine disease. And then on the right, we classified severity in a different way. And this was based on the manifestations on CT or ultrasound based on stone burden. And again, we found now a greater number of metabolites, the majority of which were overexpressed in those with a severe stone burden in the right-hand side, and then a few metabolites that were underexpressed in those with severe disease. There is a lot more work that needs to be done. We need to expand our sample size. We need to understand the stability of these metabolites in different conditions, how they would be collected at the point of care. It certainly needs to be validated in other distinct samples. And I view this as an opportunity to collaborate in a way to align our efforts to make what I think could be some really substantial changes in how we manage these individuals. So our goal, our vision, is to introduce a new era of personalized kidney stone management by reducing these barriers to testing, by going from a 24-hour urine collection to one that can be collected at home and sent in, that ultimately we hope will lead to more timely treatments and more effective treatments, and also potentially reveal new therapeutic targets that could be either targets for drug repurposing or for novel therapeutics, which are certainly desperately needed for stone disease. And that we hope will be ultimately scalable to the 40 million individuals in the US, and I need to do those numbers for Canada, but the tens of millions of individuals in North America who are living with stone disease and its manifestations. I'll end with even a further vision that is just as clear, but is going to be much longer time frame to achieve. And that's going back to the gut microbiome and the potential for therapeutics. And these are some results from a recent study where we took 5 different medications and exposed these single cultures of bacteria, or cultures of single bacteria, to different drugs at different concentrations and looked at their growth. And there were some really dramatic differences based on the type of drug and the dose of it that could potentially reveal how these medications are impacting the microbiome and how we might be able to identify not new therapeutics, but drugs that exist right now that might be repurposed for stone disease. This is so preliminary because we're looking in vitro cultures, and we're only looking at single organisms. We're not looking at the interaction with other bacteria, but through the collaborations that we have and some of, I think, the novel platforms and models, both with Dr. Lang's group and with collaborators at Penn, I think we do have the opportunity to do that. So that is where the puck is going here as a non-hockey player. And this is just an article which I'll end that was published in The Guardian that demonstrates how drug repurposing and really AI may allow us to understand through potential mechanisms of existing drugs, how they may interact with causal pathways of some of the most common conditions, of which stones is one that we have in our society. So I do think there's hope. So until we get to that point, what I will continue to do is encourage my kids to keep playing in the grass, and if they happen to get exposed to dirt, all the better for it. And I want to acknowledge my collaborators and partners in this work, of which only a few are named here. I'd like to start with Michelle Denberg, who has been my partner in this work for a decade plus. She's a nephrologist and an epidemiologist at CHOP, and having that insight from colleagues in nephrology that can complement your clinical knowledge as a urologist, I found incredibly helpful. Certainly, Dr. Lang, and our relationship and collaboration, now stretching back almost a decade, is what keeps this incredibly exciting. And I think the opportunities are even greater in the future. And then we have individuals at CHOP and elsewhere who have been key collaborators. So without a team, none of this would be possible. And with that, I want to end and just simply thank you for this opportunity. This presentation is just a wonderful honor for me. And I think even more importantly, the ability to meet new colleagues, those who will be entering the urology workforce in the future, and hopefully a time to see your impact on this field in the decades to come has been the greatest joy that I've had so far. So thank you so much for your incredibly kind invitation and attention.